Should I share my screen? Can you hear me?
Yes. Dimple madam?
Yes, sir.
Yes, please begin.
Okay. So, hello, welcome everyone. Thank you for joining us today's webinar. My name is Dimple Khambati and I am the head of Medical Engineering Department, Parul Institute of Technology, Parul University. So, today we are presenting a webinar on how Drosophila flies became my pets, the research, right, by the Ms. Ankita Banerjee. Right now, she is working as a research technician at Rahme Laboratory, Harvard Medical School, Massachusetts General Hospital, Boston, USA. So, for audience, if you have any queries, any questions during the webinar, during the presentation, please send your queries or questions to the given WhatsApp number. Right now, it's visible on the screen. We will discuss at the end of the webinar. And without taking too much time, I will hand over today's session to Ms. Ankita. Madam, please, you can start your presentation.
Thank you so much, Ms. Dimple, for having me here. I'm super excited to share my work with all of you today, and I hope everybody's doing very well. I would like to start with a small story um, about my experiences and revelations, how it helped me choose the path that I'm in today. When I was a little kid, I would often see my grandma and my mom having all the answers to any kind of health issues that we have. My grandma was a doctor, so she knew how to cure, and my mom also learned the magic of medicines and would so easily help with all our health issues. So I was deeply inspired. That was my first inspiration, the ability to cure and the ability to help. And I decided I wanted to become a doctor when I grew up. As I grew up, I used to see my dad and his ability to create. Being an engineer, he would build things up and he would create new things. And I would wonder, what if I could also create something that has the magic to cure? So, my granny and mom inspired me to the magic of health, and dad inspired me to create. I realized that it was not the profession, but the inspiration came from the ability and the idea to help, right? So, as I started to explore more subjects and courses, I developed interests in not just biology, but mechanics, thermodynamics, and I loved algebra too. So, I started asking questions like, can doctors help single handedly? And the answer was that there is a pool of professionals working together along with the doctor to make this entire process a success, right? And then I explored more and I discovered that there's a field called biomedical engineering. I pursued this field in my undergraduate, and it was from Government Engineering College, Gandhinagar. And I would like to add that I would love to say this that that is where I met Miss Dimple. She was my senior, and then she even taught me as a professor. She has been a great mentor and guided me throughout, and I can never say thank you. Like, I can never, it can never be enough. And I'm very thankful to have her and to have known her this long. After my undergrad degree, I decided to pursue masters abroad. I joined Boston University. And I have always been a very curious person. So I wanted to dive into research so that I can ask hundreds and thousands of questions. And so I did. I took research based courses here at BU. And now I'm working with Massachusetts General Hospital as a research technician. Now you would wonder, what is research all about, right? I would say it's not the procedures and the long protocols or methodologies. The core of research is asking why. why you're doing what you're doing. It's the idea of the experiment, the hypothesis, and that is what is important in research. What is the big picture and what are we trying to achieve? A good researcher is someone who can always see or predict the possible outcomes. And what are you looking for? You should always know the big picture, right? So before we dive into my work, I would like to show you how my life here looks like. I often wake up shoveling the snow in peak winters and then go to work. While returning home, get some groceries. Then the whole war begins with cooking a meal by following like 100 recipes and then trying to make sense what to mix with what. And then after that, washing vessels, and then how can we forget the laundry, right? Life looks a lot like cleaning and washing and a lot of work. But along with that, it's along with all the blues, it also includes graduating and meeting a lot of new people, socializing with amazingly intelligent people for in our field and also otherwise, even outside, right? Listening to ideas and accepting new perspectives from people all around the globe, going for bike rides with friends and making snow angels, experiencing the changing seasons here in Boston is beautiful. You can see the cherry blossom in the spring, and then leaves change their colors. They go red, orange, yellow in the fall, in the autumn, and then you welcome snow. So now my pet flies are waiting eagerly. So let's dive into and move on and let's talk science. So, my workplace is an immunology and pathogenesis laboratory. Simply put, the lab works on studying the host pathogen interactions. I will talk briefly about what I mean by the host pathogen interactions in the next slide, and that will also help you interpret our logo that I show here. Our aim is to develop alternative and novel strategies to combat and prevent infections and also potentiate host defense mechanisms. So, by unmanageable pathogens here, what do I mean, right? So, our goal is to attempt and try to design ways to fight the bacterial infections in humans, which is extremely common these days, especially in the patients and the people who get admitted to the hospitals and acquire these infections. These infections are usually called the hospital derived or acquired infections, right? So, as every hospital have different standards of care protocols, these infections are very easily acquired. But the good news is they can be avoided if taken care of. In this point, unmanageable refers to bacteria which can develop resistance towards antibiotics. Research shows that these bacterial cells transform in such a smart way that they survive antibiotic killing. Basically, there is so when you put in medicines and antibiotics, they do not die, they strive. Hence, we definitely want to overtake these little creatures, right, on their smartness and try to fight them back. So that's our first aim. Next is to avoid killing. Maybe we should try to prevent these. The point of prevention to infections is very critical here and is another very necessary perspective to look at infections. Lastly, along with understanding the bacterial mechanism, as in understanding how does the pathogen function, it is also important for us to work on how to strengthen our host response. To do that, we would need to first study how our host cells, that is the human cells, behave without the presence of a bacteria. And then what happens when a bacteria invades? So we put in bacterial cells and check that what how are the cells behaving when the bacteria is there inside the body? And ultimately, what are the different ways to combat the pathogenesis or the infection, right? So these are the three major areas of our work. Summarizing this slide, I will make it simpler and say that first, we will look at how would we prevent infections at the first place. In case we do get infected, how can we kill them or disarm these stubborn bacterial loads? And very importantly, how do we strengthen our immune response and our immunity as a defense mechanism? So there are major sectors of our research, and most important sectors that we work on are the host environment and the pathogen environment. By host, I primarily mean that it's our body, the humans, right? And we humans are the host when these microbes attack us and they try to build their homes inside of us in an attempt to survive, or maybe just like lazing around in our body, wandering around our organs, getting nutrition from the food that we eat. It is basically the unique way to survive and their designed survival tactic. In other words, host can be defined as an enclosed space which allows the invasion of these pathogens. So I'll be using pathogens a lot. I could just like use pathogen and bacteria uh, sometimes. In order to study the host processes and mechanisms, we cannot experiment on ourselves, obviously. So we found out various host models. At our lab, we worked with few of the various models that are available in research. And some of them are mice, we use and also my favorite, fruit flies. Flies are my pets now, so because I've been working with them since the start of my career, and I will talk a lot about them in the following slides. For now, let's take a look at these models. So, model is used in medical research because it can mimic, or we can say it can copy the aspects of a disease found in humans, or replicate, say. Replicate is a good word. So we can obtain information about a disease and its prevention, diagnosis, and also treatment through these models. The model plant that you see here, called the Arabidopsis plant, was the first plant to have its genome sequenced. And hence, this plant model can help us and it can facilitate rapid genetic studies in research. Whereas the fruit fly, the Drosophila melanogaster, has been extensively studied for um, genetic modeling. It also has many characteristics which make it an ideal organism because we can study animal development, behavior, neurobiology, and especially mostly it's used for human genetic diseases. um, you will see why it is the best model, I say, because in my further slides, I explain it very in detail. So you will know why it's the best model. For now, let's move on to the other very important sector, that is the pathogen environment, the smarter species. There are around thousands of different types of pathogens, and each of these pathogens have like so many strains within them. We majorly study the Pseudomonas aeruginosa. Pseudomonas is a gram-negative bacteria that can cause disease in plants, animals, including humans. I would like to focus on the word gram-negative here. Gram-negative bacteria have a thin cell wall made up of a layer of peptidoglycan. Now, gram-negative bacteria are more dangerous as disease organisms. Why? Because their outer membrane is often hidden by the slime layer and it hides the antigens of the cells. And so it acts as a camouflage. The human body recognizes any foreign body by its antigens. And if they are hidden, it becomes very hard for the body to recognize or detect the invader. So, because of its gram negative behavior, Pseudomonas is a very dangerous pathogen. It is a type of bacteria that is found everywhere in the soil, in the water, and it affects human beings. It affects it infects our blood, lungs. It can cause pneumonia and many other parts of the body. Especially, it attacks us after surgery. So, Pseudomonas acquired infections are mostly found in patients who have undergone surgery and have been in the hospital care for quite some time. These bacteria are constantly finding new ways to avoid the effects of antibiotics, and hence they become antibiotic resistance. Now, this resistance occurs when bacteria no longer respond to the antibiotics designed to kill them. They are not really killing them, and these pathogens create a different type of genetic behavior, and the antibiotics are not able to detect them and kill them. So, if they develop resistance to several types of antibiotics of different categories, these bacteria can become multi drug resistance, right? And that is a big problem. And we are facing that problem majorly these days because most of the bacteria species are multi drug resistant, and this is the major part of research that's going on now. Along with our classic Pseudomonas, we also study pathogens like E. coli, Acinetobacter baumannii, and few others in our lab. So what we do is, in summary, we take models, we study them to understand specific biological phenomena, uh, and expect to discover various model organisms to provide insight into the workings of other organisms. While researching human disease, modern organisms allow for better understanding the disease process without the added risk of harming an actual human. So that's why we have different animal models and plant models and invertebrate models. We study various mechanisms of infections by infecting the host model with the pathogen we're interested in. So it could be Pseudomonas or it could be E. coli, depending on what aim we have and what insights we're looking for and what infection we are looking for. And we work on designing also various inhibitory compounds, majorly for drug discovery against the multi drug resistant pathogens. Okay, so let me introduce you to my pets now. This is one of the thousands and thousands of fruit flies that I have. Let's dive into details. I'm so excited to talk about, and you can then see some of my pets too. Here's an overview of Drosophila melanogaster as a model organism. Drosophila melanogaster is a scientific name for fruit flies. These are the same regular flies that you see buzzing around your bananas and other ripening fruits at your home, your office, at the marketplace. Drosophila feeds and breeds on fermenting fruit or any other sources of fermenting sugar, sucrose, fructose. They're attracted to sugar, sweet stuff. These flies are widely used model organisms for scientific research. By studying this organism, it provides us extensive insight into eukaryotic cells and genetics and human disease. As I mentioned before, the fruit fly has been extensively studied, and it's the best model organism for genetic investigations, which I will uh, dive deep into in my next few slides because it has a very simple genetic framework and very less genes. So I will talk about that more in my next slides. Um, and These flies are used to study, as I said before, animal development, behavior, neurobiological diseases, neurodegeneration diseases, and many other genetic diseases, immunocompromised diseases, etc. Now, the next big question that you might be pondering on would be why is Drosophila such a powerful model? So, these are the points. It is small. They are so tiny. Due to the teeny tiny size, they're extremely easy to handle and anesthetize. So, we use carbon dioxide gas for fly anesthesia. I will show you in one of my following photos what we use for anesthesia and how we do that. It has, so we use very inexpensive and very cheap equipment. So the equipments that we use to maintain these flies and house them basically in the lab are very affordable, very cheap. We use these, I've shown the picture here. We use these plastic vials and we make the food and just put a cotton and that's all. And you just like maintain these flies, grow them, culture them. You can do everything pretty much very cheaply. They have simple genetics. So the Drosophila genome is comprised of only 4 chromosomes with approximately 14,000 genes. This makes studying these flies very, very easy, very exciting, and less complicated, right? Mice has like so many genes and it's very complicated, but with these flies, it gets very easy. They have very limited genetic redundancy. So, to explain this point clearly, I would take an example of a mice. So, mice may have like 3 copies of a gene causing a particular phenotype. So, by phenotype, I mean any physical behavior or like a physical indicator. So, when one gene is mutated, the others can compensate, leading to no observable developmental or physiological change. Say, for example, you want to change an eye color. So the fly would just have one single gene, but mice can have 3, 4 genes that compensate for the other one. So if you mutate one of the genes showing that physical behavior, you can't see the change because there are many others which also show the same physical change. Thus, the mutagenesis experiment in mice is less informative. In contrast, flies may have just one version of the gene, as I explained. So, basically, pulling everything together in simple words, we can say that you can attain observable differences in the genetic outline in a fly very easily in comparison to a mice. They have short life cycle. It takes approximately 2 weeks from when mating is set up to generate a new adult progeny. Females are extremely fertile and they can lay hundreds of eggs per day. And experiments with flies can be conducted quickly and with very high sample numbers. So you can get a lot of flies very quickly and you can um, study generations in a very short period of time. They have genetic similarities to humans and other mammals. So, like approximately 50% of fly genes are similar to mammalian or human genes, meaning the gene originates from the common ancestor. There's like 75% of human diseases that relate to fly genes. So they have orthologs and similar functionality genes. So can you imagine Drosophila and us, we are so similar and we never knew that. So this makes Drosophila a great and extremely useful model organism. Let's get to know Drosophila. Okay, so Drosophila have three main body segments: the head, the thorax, and the abdomen. A single pair of wings and three pair of legs. They are between 2 to 4 mm long and weigh about 1 milligram. Females are typically larger than males, and the wild type fruit flies have large red eyes and pale yellow or light brown bodies with black stripes on the abdomen. The relationship between fruit fly and human genes is very close, as I said. Often, the sequences of newly discovered human genes, including diseases like human disease genes, um, they can be matched with equivalent genes in the fly. So, like, who would have thought that these little creatures could resemble us with so much depth, and we have no idea about it? We just shoo them away from our kitchen bins and fruit baskets, but these are us. So, the Drosophila life cycle is about 2 weeks long, as I said, and it comprises of four major stages: the embryo, larva, pupa, and the adult. Drosophila larvae are small, white, and glossy, with similar appearance to worms. They look like worms. And Drosophila can survive in room temperature in a range of like 12 to 35 degrees Celsius. In the lab, we have incubators set to 25 degrees and 60% humidity. A female may lay eggs up to like 30 to 50 eggs or hundreds per day throughout her lifetime at room temperature. And daily egg production is reduced if you put them in lower temperatures. So, in case we want to maintain the fly stocks for longer span of time, before we change the food, we put them in cooler temperatures. So, like during COVID times when we couldn't go to the lab and we had to maintain them because they need, even though they're very easy to maintain, they need regular maintenance. Like you need to look at them every day, take care of their food, and flip them each day, each week. So what we did during COVID times was we would keep them in cooler temperatures and so that we don't have to go very often. So the cooler temperature reduces the egg production. So it prevents the fly overcrowding. So lesser the eggs, lesser flies in the vials. And if each vial have less flies, it will have lesser chances of overcrowding. And if it's overcrowded, the flies can die. And if they die, we cannot. So we have different fly lines in the lab, and each fly line is having different kind of genetic design. That so we have transgenic flies in our lab, right? So we have different mutations done in each fly line. So they have the same genetic framework, but we have different mutations there. So we need to take care of all the fly lines, and we can't afford that they can die, right? So Drosophila have a short and very simple reproductive cycle, and it is normally about 8 to 14 days, depending on the temperature, of course. This means that several generations can be observed in a matter of months. That's the most important plus point for us in research because you need a high sample number and you need to check generations of flies in very less time so that you have more number of experiments and you can repeat those experiments. So these fly models help us conduct experiments and get observable results within months' time. So the average lifespan of Drosophila is between 60 to 80 days. However, lifespan can again get affected by factors like temperature, overcrowding, but it's roughly 60 to 80 days. So You might ask me, how do we tell between males, females, and virgin flies? By virgin flies, what we mean are the newly born, extremely young flies prior to their mating life stage. So to distinguish between the gender, we need to look at the species very carefully under a microscope. On the left side on my slide, you can see that the fly at the left is a male, and it is significantly smaller in size to that of its female counterpart at the right. One more very important distinction factor is that the males have a darker abdomen, as you can see. The male's abdomen is rounded at the bottom, while the female's abdomen is pointed. It might be a little difficult to notice right now, but as and when you keep looking at them and observing them really close, you can actually differentiate very well. The males have a dark circular apparatus, the penis apparatus, at the end of their bodies, while the females lack them instead and have a pointy end. On the right hand side section of the slide, you can see the virgin flies. In genetic research, the most important part is to look for virgin female flies so that we can make the required crosses. Now, this would confuse you, but I will explain you more about the virgin females in my next slide. To find these virgins, we need to look for flies with immature wings. So as you can see, the first picture here shows us immature wings. That shows that it just came out of the pupa. Also, the virgins are very transparent and much bigger and bloated compared to the darkened adult flies. So here I show the bloated and transparent. It's pretty big. And the other one shown is a non-virgin fly, which is darkened, and so it shows that that's an adult, like much. So that fly came out of the pupa much later and have lived his a little bit more life than the other one. Moving on. So these are some of the pictures of my fly stocks at my workplace. The image at the top left corner shows some of the trays with collections of my Drosophila fly stocks. The two images at the bottom are captured to the eyepiece of a microscope. Those are females too, but those are adult females because you can't see that they are like transparent or something like that, right? But yeah, it's not clear very well, but yeah, those are microscopic view. And on the right is a video of some of the transgenic flies that I work with. We flip different fly lines into different vials with the same food. The food comprise of corn, agar, yeast for nutrition, and of course sucrose, which is the major ingredient. So they get attracted to sucrose and sweet stuff. They mate in these vials and also lay eggs. So then you can just collect the progeny as and when needed. So this is the fly anastasia pad that I was talking about. This pad has a continuous supply of CO2, carbon dioxide. So the moment you flip the flies on the surface of this pad, they get senseless and anesthetized. And then we can look at them under the microscope, find males, segregate females, collect virgins, and dissect them as per your requirements. So few slides earlier, I mentioned that virgin flies are of great importance to us in research. You would wonder, why do we care about virgin flies? The answer is we need female virgins for our experiments because they will produce offspring of interest. Depending on the type of genetic line we want them to express. Okay. So Once the female flies emerge from the pupa cases, they have a certain number of hours before they will start mating with their brothers or other males in that vial. If you cross a non-virgin female, so by crossing is you put males and females together and they will mate. So that is called cross. So if we cross a non-virgin female with a desirable male in a new vial, the non-virgin female will produce some offspring that are not from the desirable males, which can really mess up our results, and we don't want that. So what we do is we take virgin flies and put it in a different vial and keep them separate. So they don't get to meet males. And then whatever kind of fly line males we need. So these are the males which have mutations done in their genetic framework. These are the transgenic flies. So we take those males and put them in the virgin female vial, and there you get the crosses. And after they mate, after like 5 days, you take out all the adult flies and you just have the eggs. So after some time, you just have an empty vial with eggs, and then you get the progeny flies, the new flies, and they are totally different. And I will explain more about how this happens in the next slides. So I have a short video here showing how we look for virgins and how we collect them. Before you watch it, I would also add that virgin flies have something very unique along with their transparent body. They have a black spot, a black dot, which is visible through their abdomen. Um, that dot is called meconium. The meconium is a dark area that we will see here in this video, and it is the remains of larval food. So it does not take a long time for a fly to age. And after the fly hatches out of the pupa, in like 4 hours or so, it becomes more difficult to tell the difference. So even though the fly is a virgin, it will darken. So its body will darken up. So then it will get very difficult for you to tell if they are a virgin or not. We don't take those flies. We just don't take chances. We just take very young flies, and which you can actually see that they are transparent and you can see meconium in them. So this video will show. So these flies are on the anesthesia pad that I just showed you, and you just anesthetize them, and you can look really closely. So what do we do with these flies after we mate them and get them segregate them? After so there are various methods of activating gene expression. So we need these flies for genetic mutations and genetic studies. So there are various methods of activating gene expressions in Drosophila fly models. One such method is the UAS GAL4 system. I will try to make it very simple. So we use transgenic flies with mutations generated using this system. So within the system, you can decide how you want to play with the genetic expression. So you can either want an overexpression of a certain gene or a knockdown, as in a reduction of genetic expression, right? So depending on your needs of the experiment, the transgenic flies are generated. Here you can see the two possibilities, an overexpression or a knockdown. In our lab, in the projects that we work on, we use knockdown flies, which basically mean we want to suppress down the expression of the gene of our interest. And hence we are using knockdown flies. So to obtain knockdown, under the US GAL4 system, we use something called RNA interference method. Um, RNA interference, or RNAi, is a type of gene silencing mechanism. In that mechanism, what happens is there's a double-stranded RNA molecule that directs the specific degradation of the mRNA, of the corresponding mRNA that's produced. So this method, when used in combination with the UAS GAL4, leads to a reduction of the expression of a certain gene instead of overexpression. Okay, so let me explain the US GAL4 system a little briefly. So as you can see here, the GAL4 consisting flies are termed as the driver strain. So I have it here. So you see there is a driver strain and the responder strain. So the GAL4 is called the driver strain, and the UAS containing flies are called the responder strains. Now, geneticists have created genetic varieties of fruit flies. There are many varieties, and some of them are called the GAL4 driver lines, and they express GAL4. So, an important part is some fly lines might express GAL4 only in muscle cells, or only in nerves, or only maybe in antenna, or as it is shown here, in the wings, and so on. So the GAL4 gene is placed under the control of a gene promoter or a driver gene. Now, the presence of GAL4 by itself in these cells has very little or no effect at all, since the GAL4's main effect is to bind to the UAS region. The most cells have, and most of the cells do not have UAS. So basically, what you want to do is you want the GAL4 to attach to the UAS so that the corresponding gene can be expressed. So the other necessary part of the system is the reporter lines. These are the UAS strains, the strain flies with UAS next to the desired gene. Now, these genetic instructions occur in every cell of the animal, but in most cells, nothing happens since the cell is not producing GAL4. In the cells that are producing GAL4, however, the UAS is activated. So the gene next to it is turned on and it starts producing the resulting protein. So what you're doing is you get to choose the tissue type with the driver flies, that is the GAL4 flies, and the gene of interest with the responder US strains. Therefore, this is exactly how we define the areas we are interested in according to our project aims. So we can decide what tissue type we want. So we express GAL4 in those tissues. And what genes do we want them to express? We use the UAS, the responder strains. So at our lab, we're primarily working on two broad projects with Drosophila models. First one that I show you here is the aging project, where we use the gut or the intestine samples from the flies to carry out our experiments. The second one here is the Neurodegeneration Project. This one deals with the fly brains and studying them to uncover some mechanistic pathways for Parkinson's disease, primarily. A mechanistic understanding of the natural aging process is very essential for developing interventions in an attempt to extend health span. So, what we try to do is we're trying to examine a protein called heterochromatin protein type, which is found in intestinal stem cells, and it plays a major role in the aging process. So, the Drosophila model helps us in studying aging and also neurodegeneration related diseases like Parkinson's disease. And here in this slide, I just wanted to show how similar mammalian intestine is to Drosophila intestine. How like it's very similar, and you can make amazing use of Drosophilas this way. Also, the brain is very similar. Very briefly, I would go over these two projects. What we're trying to achieve through our aging project is to challenge the current approach towards aging related diseases and propose a new trending approach with a very different perspective. So, previous therapeutics strategies focus on aging related disease at a time and after. So, they would attack each aging related disease one by one and try to suppress them. But in contrast, we propose targeting aging. So we say that if you target aging, it can prevent and also delay the onset of the multiple array of aging related diseases. Therefore, it becomes a very important alternative approach. And Drosophila helps us do that. Likewise, through our neurodegenerative fly models, we aim to see changes in the knockdown flies, and they replicate the behavior of neurodegenerative human brain neurons in the very common diseases like Parkinson's. So we see that the knockdown flies have their motor functions affected, which happens in humans too, and when they undergo neuronal damages. So we see we see great results. So wrapping up, I would like to share some of our findings through the knockdown flies. We see reduced motor functions, hormonal imbalances, fluctuations in neurotransmitters, and increased inflammation in the brain of the transgenic knockdown flies in the brain project. While in the aging project, we see accelerated aging phenotypes and shorter lifespan, metabolic imbalances. So the idea is if a knockdown can show us all these things, what happens if we overexpress those genes? So maybe we can combat these problems, right? That's the whole idea. So I'm along with the flies, I thought I would also briefly talk about one more very recent technology we're working with at our lab, and that is the Organon chip technology. Organon chip technologies, they seek to replace animal models and cell culture by allowing us to study human tissue with a 3D structure perspective. And it also has microfluidic channels, and it's basically a way to mimic the normal physiology of human beings. They're very easy to use and they're decently affordable. This could potentially speed up the drug discovery process in many ways. Basically, what these chips do is they're meant to replicate the human body and human body's internal environment. And these chips are then put into a system which provide them with stretching, like you would have peristaltic movements in your stomach and cardiac, systolic, diastolic movements, etc. Also, these systems provide proper pressure and temperature conditions, as you would expect in a human body. The idea is to provide the exact same body conditions and continue the research with more precision. So, I have a very, very cool video for you to have a look at. Yeah. So, here in this slide, I'm showing the major elements of a human emulation system. So, along with the chip, there are three very important devices that I show here. Those are the pod, the Zoe, and the orb. The pod is the portable module, and the chip is attached to the pod. The pod provides a supply of media in a steady manner, and it gives nutrition to the cells in the chip. The Zoe, whereas, is the culture module. The Zoe controls the rate of flow and gives stretch to the chips, just like peristaltic and cardiovascular um, systems, providing the environment to the cells that they need to live. Zoe provides with all the mechanical forces, basically. The Zoe is further fed with carbon dioxide by the orb. And orb is the hub module. It provides gas and vacuum, which is required by the Zoe to function. So we are collaborating with this amazing startup, Emulate, which came out of Harvard School, and they are doing a great job on Organon chip um, devices, and we have one in our lab. So this is one of the images of the pods along with the chips. The chips are inserted under the pods, and they're connected. So this is all these pods have media. So you put in media, and the media goes into the chip. The chips have human cells. So you see it in human cells in the chip, and then you connect it to the pod. The pod will have media. The media will go into the cells for them to survive. And then you put these pods inside the Zoe, as I showed you. The Zoe would give continuous pressure, temperature, and stretch. And then this is how they have designed just to mimic the human body. So this is just a schematic of the chip. This is an intestine chip model. These chips are designed uniquely. So for each of the organ types, there are like 7 to 8 types that they have made, and I think they're still developing new types. They have liver, they have kidney, heart, even have brain. Um, so you can seed in appropriate cell types into these channels and then carry on with your experiments. Working with this system was very interesting, and I personally loved to get the hands-on training on this one. And I've done intestine chip majorly, and I have some pictures that I can show you. So here are some of the images that we captured with the intestine chip model. You can see the epithelial cells and the endothelial cells, and how they look under the microscope. And this is separately. So they are growing without connecting. Below, you can see that after seeding these cells and letting them grow on the porous membrane that is in the middle of this chip, you will notice that the cells interconnect and they form some villi-like structures, right? So this was really very interesting to look at, and the ability of this tiny chip to have so much scope in immunology, immune response. You can put in bacteria, just like the video showed, and you can study how the bacteria reacts to the cells and how the cells behave. Do they shrink? How does the white blood cells react to the invasion of pathogens? So this technology is really cool, and it gives amazing precision. So basically, the human organ chip helps with the three major areas, and those are basically disease modeling, drug testing, and mimicking organ functions. Clinical studies take years to complete, and testing a single compound can cost more than like billions, right? So, and also while using animal models, and animal lives are lost, and the process often fails to predict the human responses because traditional animal models often do not accurately mimic human pathophysiology. So like mice is a big question now. It's in deep it's like a debate now whether experimenting with mice actually mimic the human pathophysiology. So for these reasons, there is a broad need for alternative ways to model human diseases, um, in order to like fasten and accelerate the development of new drugs and advance personalized medicine. So these models are very helpful. So I would like to wrap up, and I would like to add, no matter where you study, which organization you work for, what you do, how you do it, the only thing that really matters is who you are and what you believe in. Believe in yourself, and you will be fulfilled and you will feel successful in everything that you do. I feel immensely grateful and happy now that I'm able to be with you and give this talk. And I hope this talk succeeds in inspiring some of you. I hope it sparks a light of interest in you towards research or science, or even if you learn something new today, I would be extremely happy. We are all researchers in our lives, and because we are all curious and constantly trying to figure out our calling and purpose. So keep on it, treat yourself with compassion and love every day. And I would like to leave you with one more thing, one quote that I love immensely. Take risks in your life. If you win, you can lead, and if you lose, you can guide. Thank you very much.
Thank you, Ms. Ankita. So we'll go ahead and take some time for the questions.
Yeah.
I have received one question from the student.
Uh-huh.
In Drosophila, germline cells are located.
Sorry, you're cracking, Miss Digital.
Yes, in Drosophila, where the
Mhm.
germline cells are located.
Germline cells.
Yes, yes.
So
I'll write in the chat box.
Yeah, so the germline cells and the developmental stages, you can actually, so those are called instars. So when the pupa grows, there are instar developmental stages, and you can get the developmental genes from there, and you can develop transgenic lines by using various bacterial lines. So basically, what they do is they put some bacterial cell lines into the development genotypes, and then what they do is they cut the genes and replace it with the desired genetic outline and whatever genes you need for the experiments, and they mutate it that way, if that answers your question.
Okay. The another question is, I'll write in a chat box. Does using ethanol for the de-waxing Drosophila heads before dissection interfere with future protein extraction or flying prey imaging?
I'll check the chat. So it's a debate actually. So we do not use ethanol for dewaxing Drosophila heads. And it is actually a very good question. It does. So there are various research papers on this, and ethanol does affect, it seems, but we do not use it. So um, we just anesthetize them, dissect the flies, and get the heads. And if you want to dissect the brains, we use PPS. We just use that. We do not use ethanol for any of our section or even anything. We do not use ethanol.
Okay. So there is no more questions.
Thank you
So
so
if you
much.
want to give some, if you want to give some suggestions, ma'am, then you can please suggest to students.
You can ask any questions if you have related to this, or any you would like to get some any advice. I would be happy to answer any questions. You can reach out to me on LinkedIn. And all the best. Thank you
Great.
for having me here, Ms. Dimple.
Thank you, everyone. We appreciate you being here. Thanks to Padu University for giving me the opportunity to host such a webinar.
Yes,
Thank you,
thank you
madam.
to Parallel University from me
Thanks
too.
again for joining us today. And we
Thank you,
will see
thank
you
you.
next time also.
My pleasure. Thank you. It was fun.
Have a nice day.
You too.